All right, I'm on. So I can't possibly go over every single word or detail about this nervous system that your book describes. I just simply don't have time. Even if we were live in a room together, one-on-one, and I was lecturing to you live in person, I still wouldn't have time to do it. So I'm going to try my best to hit as many topics as possible and explain them in plain speak for you. So I'll call it my first whiteboard, and I'm going to start making, so we're just talking about the nervous system. So here's my header. Just so you know, this system regulates the body, regulates homeostasis in the short term. It's super fast relative to other regulators like the endocrine system, which tend to be quite slow. So for short-term regulation, this is what our body uses almost exclusively to regulate things within physiologically. There are three functional elements of the nervous system. And there are also sort of two major anatomical divisions. Sorry, that's supposed to read anatomical. So I'm going to briefly talk about these first. We just categorize the nervous system, and this is largely anatomical categorization. The central nervous system, and from this point on, after, that's supposed to be an R, Nervous, which includes the brain and spinal cord. From now on, I'm just going to say and write CNS. And by writing or saying that, I mean central nervous system. And the anatomical structures are the brain and spinal cord. So we find those in the central nervous system. And then the second division is called the peripheral nervous system. And I will say and use the abbreviation PNS, just so that we can get through this before midnight tonight. And the peripheral nervous system anatomically is made up of what we call peripheral nerves, nerves emanating mostly from the spinal cord, but also a little bit from the brainstem. So I'm just going to write nerves from the spinal cord and brainstem. So that's the anatomical division: CNS, PNS, central nervous system, brain, spinal cord. Peripheral nervous system is basically everything outside of the brain and spinal cord, which are just peripheral nerves. Now, these divisions, before I talk about, just put this on hold for a minute, three functional elements. Before I get off of the two anatomical divisions, I'm going to add what each of these, the central nervous system, is largely involved with what we call integration and processing. We call it the control center. So I'm just going to write those two words, integration and processing. And the peripheral nervous system is mostly, functionally, the peripheral nervous system is an information highway. It's a transmission, I'm going to write transmission pathways. Now I'm going to talk about the three functional elements of the nervous system. I'm just going to call up a new whiteboard in order to do that. Three functional elements. And this is very easy to understand. It just might be new language, that's all. We have first sensory input. And this is sensory receptors that sense a change in the environment and send an impulse toward the CNS, central nervous, the brain and the spinal cord. Then we have something called integration, and that's what happens in the central nervous system after the sensory input signal arrives to the central nervous system. Integration, this occurs in the CNS, the brain and the spinal cord. And that's the processing of the sensory input. So I'm going to use this word, processing of the incoming information. And then there's some sort of decision about what to do about that incoming information. And then the sending of a command or some sort of output. So we get processing, let me erase that comma really quickly. Processing of input. And then I'm going to just write decision, consciously or unconsciously, subconsciously. So we get processing of input, decision, and then the sending of a response or command. That's largely what goes on in the central nervous system. And finally, after that command is sent out, that response is sent out of the central nervous system, the functional element is called the motor output. And the motor output is simply the response of the command to a destination. And we call that destination an effector. And that can be pretty much muscles or glands. Now, muscles can be skeletal muscle, cardiac muscle, smooth muscle. It doesn't have to just always be skeletal muscle. So that sort of gives us an idea of the anatomy of the nervous system. And the anatomy is this. Here's the brain. There's the cerebellum. This is a lateral view. There's the spinal cord. And then we have nerves, and they usually emanate off the spinal cord in right and left pairs, like this. And those nerves go everywhere in the body, to our visceral organs on the inside, to our muscles, to our glands. And then there are nerves as well, peripheral nerves, also emanating from the brainstem, going to and from things in the brain as well. So that's the anatomical view. And then functionally, we have an incoming signal and then a processing area making sense of that signal and then issuing a command. Actually, I should have drawn the incoming signal to the brain and then the issuing of a command out of the brain. So it gets sensory input, integration, motor output. And the nervous system almost always works like that. So I'm going to draw a quick flow diagram, and I'm going to write sensory, afferent. The word afferent is just a directional word. It means toward the central nervous system. And the sensory receptors in our bodies that set up here, that sense a change, can be either on the inside of our bodies, we call those visceral receptors, or sensory receptors that sense a change in the external environment, we call those somatic. Okay? They send a signal, they sense a change, and they send a signal to the central nervous system, the brain and spinal cord. And then it's in the brain and spinal cord that incoming information is processed. And then if a decision is made in the central nervous system that something needs to be done about this sensed change, then an output command is sent. We call that the motor efferent with an E. That just means taking a signal or sending an impulse away from the central nervous system to effectors. There's two subdivisions of this motor efferent division, functional division, and that we call either somatic or autonomic. Somatic responses, commands, somatic commands go strictly to voluntary skeletal muscle. So I'll give you some classic examples of these pathways as soon as I finish writing this. Autonomic, almost think that it's spelled automatic, because the autonomic go to things that we don't have conscious control over, like cardiac muscle. So I'm going to write cardiac, smooth muscle, think digestive and respiratory organs, and glands. So the effectors of the somatic motor division are voluntary skeletal muscles. The effectors of the autonomic division of the motor nervous system are cardiac muscle, smooth muscle, and gland. Now let's do some examples. I'll use green for my first example. Let's just say you stick your finger on a hot stove. There are sensory receptors in your finger, in the skin of your finger, that sense that dramatic heat, that temperature change. And those receptors fire an electrical impulse, and they send that impulse to the brain, to the central nervous system. In the central nervous system, the brain processing is done. The brain says, heat. We better do something about that. If we don't, we're going to have tissue damage and burn skin. So then the central nervous system sends a command out via this motor efferent pathway, this direction, to the skeletal muscles in our arm and hand to pull our hand away from that heat really quickly. So that's the pathway. Sensory, send, processing, send another command out to an effector. In this case, it's voluntary skeletal muscle to jerk our hand away really quickly. That all happens in less than a second. It happens in milliseconds, super fast. Now let's take one more example. I'm just going to arbitrarily pick red so that you can see it. Let's say you just had lunch or you just had breakfast and you swallow the food and it makes its way to the stomach. Now something has changed on the inside of your body. There are sensory receptors, this time they would be visceral, in the wall of your stomach. There are sensory receptors. There's stretch receptors there, as well as pH receptors that sense a change in the environment in the stomach. And they send a signal to the brain stem, which is part of the central nervous system. And the brain stem says, food and stomach. We need to do something about that. Let's start digestion. Let's start breaking it down. And so the brainstem then sends a signal, a command via the motor efferent pathway to smooth muscle cells in the wall of the stomach. That starts the stomach churning, the smooth muscle contraction. Also, there is a signal sent to the glands in the wall of the stomach to start secreting things like enzymes and hydrochloric acid to break that food down. So those are two classic examples, and the nervous system always works that way. It always starts with some sort of sensation, change either in the internal or external environment, then receptors sense that change, send a signal toward the central nervous system, which processes that information and sends a signal either to our voluntary skeletal muscles or to our involuntary things like cardiac, smooth muscle, and glands. If you can keep that sorted out in your mind, then some of the detail of this will be more easily understood. All right, time to move on. I'm going to talk about, I briefly talked about these two subdivisions of the peripheral nervous system. And I'm going to write some of this stuff out for you as quickly as I can because I need to cover a lot of material today. So within the peripheral nervous system, I'm just going to write PNS. That means everything outside of the central nervous system. There are two, I listed this in my diagram, but I'm going to write it down. There are two functional subdivisions. I'm going to list them and I'll say a few words about them. First, there is a sensory afferent division, and you can refer back to that flow chart that I made. Whenever you see the word sensory, always hook it up with the word afferent. Just remember that this functional subdivision always carries sensory input toward the central nervous system. Okay. Now, we had two subdivisions that I listed of this. We had something called somatic, and they're somatic sensory fibers. And these are fibers from the skin, skeletal muscles, things like joints. And then I wrote the word visceral. I'm going to tell you right now, these monitor the external world, the external environment, and these monitor largely the internal environment. So visceral fibers, I'm just going to write from organs. Mostly in our abdominal cavity, but also from our thoracic cavity, like respiratory organs as well. So we have a sensory afferent division and a motor efferent. Efferent just means taking impulses away from central nervous system. And there are divisions of this as well. They have, some of them have similar names, but when we get to the autonomic, you'll see that it's different. So I'm going to write A, somatic, and we already listed this in our flow diagram. These are impulses, commands that go to effector organs such as voluntary skeletal muscle. And then the autonomic subdivision of the motor efferent, I'm going to write cardiac, smooth. That's cardiac muscle and smooth muscle and glands. We don't have conscious control over this one. It just seems to happen without us having to think about it. Now, to make things even more confusing, there are subdivisions within the autonomic nervous system. And those two subdivisions are called, you might have heard these words before, sympathetic and parasympathetic. I'm not going to go into the details of these right now. Sympathetic, always think fight or flight. It's a survival response when we are really stressed out. Parasympathetic, always think of rest and digest. It's a maintenance part of the autonomic nervous system that we don't have to think about. My example of eating food would be regulated by the parasympathetic division of the autonomic nervous system. If you were running for your life, running from a fire or a bomb or fighting for your life, those actions would be regulated by the sympathetic nervous system. Okay, let's move on to a new topic. I'm going to talk about nervous tissue. There's basically two cell types in the nervous system. Those would be neurons and neuroglia. I'll talk about neuroglia first because I think your book does, but let's do just a quick right. There are neurons. These are the money cells. These fire an electrical impulse. So I'm just going to write the word impulse. That means action potential or electrical impulse. I'm just going to give you some characteristics. These cells, the neurons, our nerve cells, are amitotic. That means they do not undergo cellular division. What that means is that we can't make new ones. So we are born basically with the billions of neurons that we have, we're born with that number. And once they die or get damaged, they're not coming back. But the good news is they have a very long lifespan. And that's good because they don't replicate or divide. And they also have a super high metabolism. So we find lots of mitochondria in there to make ATP, lots of ribosomes to make proteins. The second type of nervous tissue or nerve cells are called, it's a whole class of cells called neuroglia. Neuroglial cells do not fire electrical impulses. These are largely considered support cells to the neurons, the electrical firing cells. So I'll get into the different types of neuroglia presently, but I did want to say that the neuroglia, they are mitotic. They can replicate or divide. So when you hear of somebody, for example, having a brain tumor or brain cancer, it's largely a glioma, which is a cancer of these cells, an uncontrolled cellular division of these neuroglial cells. And they're highly metastatic as well as highly malignant as well. All right, now I'm going to talk about the specific types of neuroglia, and I'm going to just hit on them as quickly as I can because, frankly, there's 6 different types, 4 in the central nervous system and 2 in the peripheral nervous system. So I'm just going to write CNS, and I'm going to list them 1, 2, 3, 4. I'll probably say more than I write just to save time. First type of neuroglia are astrocytes. They get their name because they're star-shaped. That's hence the name astro. These are largely, their main function is structural. They provide structure for the nervous system in general. They help anchor neurons. They help form synapses, which are the junctions between a neuron and some other cell. So they have this huge structural capability. They also help maintain something called the blood-brain barrier. Probably, if you haven't heard of this, if you have heard of it, then maybe you have an idea of what this is. This is a physical net-like structure. So these astrocytes, they have processes coming off of them, and they make this meshwork, this net, that allows some things. Let's just say this is neural tissue here of the brain, and this is a blood vessel. Astrocytes allow some things from the bloodstream to get to neural tissue, and other things can't. So it provides this barrier. It's not a 100% complete barrier because our neural tissue does need things like nutrition, but it does help the blood-brain barrier, this net, this meshwork helps keep out bad things like bacteria and viruses. It's not 100% effective. These cells also have a recycling capability. So I'm going to say they recycle neurotransmitter. That's Neuro. To remind you, neurotransmitter is the chemical that's secreted by neurons. And they do other assistance functions as well. They help with brain function. It's theorized things like memory and learning. Let's move on. I'm going to go to number 2. I'm talking about neuroglia in the central nervous system. So next page, I'm just going to write a number 2 after astrocytes. I'll go in the, I think I'm going in the order of your book. If not, that's not important. Microglial cells. Micro means small. So these are very small cells that have these thorny processes coming out from them. Mainly, their main function is that they are phagocytic. That means they have the ability to phagocytize other cells. That means engulf or ingest or eat. They act like immune cells, like macrophages. I'm going to write immune cells. So if something bad should get into our nervous system, bacteria, viruses, or even waste products, these microglial cells help clean it up. Number 3, ependymal cells, EPENDYMAL. These are cells that have cilia on them, those extensions that look like little hair processes. And what that cilia does, so we find these cells, first of all, lining brain cavities in the central nervous system, and around the spinal cord. The cells kind of look like this. They have these cilia, these hair-like structures, usually on one side of them. And if we line a whole bunch of these up together, and if this is a fluid-filled cavity on the cilia side here, the beating of those cilia, those hair-like structures, help circulate the fluid. So I'm going to write, these cells help circulate cerebrospinal fluid. Number 4, last one in the central nervous system. Remember, we're talking about support cells of the nervous system. It's a long, funny name, but they're not very complicated. Oligodendrocytes. These have the main function of creating what's called the myelin sheath in the central nervous system. That's a form of insulation that wraps around the axons of neurons. So they look sort of like an octopus. So if this is the cell body, they have these long flat processes that can wrap around neurons or nerve cells and help insulate them. So that the myelin sheath is just a form of insulation wrapping around a neuron. So I'm just going to quickly draw, this would be the process of a neuron. And this green stuff by the oligodendrocytes helps insulate this. It forms a wrapping of insulation around this neuron. These are all, just to remind you, 1 through 4 are in the central nervous system. The last 2 are in the peripheral nervous system. So I'm going to go to this. We're talking about neuroglial support cells now in the peripheral nervous system. And there's 2 types. So I'm going to write 1 and 2 again, just so you don't get them confused with the first 4. Satellite cells. They are somewhat analogous to astrocytes. They have, their main function is structure. All right, structural support. That's supposed to be an a U. I started to write my P before I finished my U. So they provide largely a structural support in the peripheral nervous system, and they help form synapses, just like astrocytes do in the central nervous system. Remember, a synapse is just a junction between a nerve cell and another cell. Second and last type, SCHWANN, Schwann cells. These are lipid-based cells that form the myelin sheath in peripheral nerves. That's a form of insulation, and it's fat-based myelin sheath in peripheral nervous system. These behave slightly differently. If this is a nerve fiber, an axon of a nerve in the peripheral nervous system, Schwann cells literally wrap around the axon in chunks, like a jelly roll, like this. So if you were to take a fluid-filled Ziploc baggie, put some water in it, and then wrap that fluid-filled bag around, say, a copper pipe or a PVC pipe, that's sort of the same, that's mimicking what's going on. And they literally wrap around in segments like this, these Schwann cells, and they form what we call the myelin sheath. Now I'm going to go to, that's it on neuroglia. I'm going to draw, we're going to switch gears and talk about neurons. These are the money cells that fire electrical impulses. So let me go to a new board. What I'm going to do is I'm going to draw a typical motor neuron. It's the classic example that all textbooks use. And I'm going to show you the anatomy and talk a little bit about the parts and the physiology. So I'm going to quickly draw a typical motor neuron. And I'll label the parts for you and talk about their function. The large portion, this is a nucleus while I'm finishing up my other pieces. This is the nucleus, and it resides in that large surface area place called, that would be the cell body. It's got a scientific name called the soma. So this is, I'm going to label this the nucleus, and the nucleus is found within the cell body or soma. I'm not going to draw all of the organelles found in the soma, but just to iterate to you, there are typical organelles in there. Things like endoplasmic reticulum, both smooth and rough. There's Golgi apparatus in this part of the cell. There are tons of mitochondria, ribosomes, all that business. I'm not going to spend my time drawing all of that in there. I'm going to highlight what's special about this nerve cell. These branches that I've drawn off of the cell body. Another name for a branch is the word process. So process is used confusingly, in my opinion. That's a verb when we talk about the brain processing information, but it's also, in this case, a noun that means a branch or long, thin segment. So these processes, I'll use a different color that I've drawn here and here, are called dendrites. Dendrites receive impulses and send those impulses toward the cell body. Okay? This long process out here is the axon. I'm going to say this very slowly because it's critical you remember this. All nerve cells, all neurons only have one axon. So I'm going to write the number 1. Now, different types of neurons can have one or many dendrites. So this is variable depending on the type of neuron. This is absolute. There's always one axon. Now, this part of the axon that emanates from the cell body that's thick right here is called the axon hillock. And it's where the axon first emanates from the cell body. Why is that a big deal? Well, functionally, the axon hillock is also what we call the trigger area. That's where an action potential, an electrical signal, is first generated. All right, a few more things about this. If we myelinate this axon, by the way, dendrites are never myelinated. Axons are often myelinated, but not always. So I'm going to use green, and I'm going to draw some myelin around this axon. So I'm creating what most books call the myelin sheath, because it encircles the axon. And this is, if this were a peripheral nerve, that would be Schwann cells wrapped around this axon. If we were in the brain, it would be from oligodendrocytes. There are gaps in this myelin sheath. So let me label this myelin. Those gaps that I've left in the myelin sheath are called nodes of Ranvier. Right there. I'll pluralize this. I'm going to write nodes. And I'll point to another one right there. That'll become significant later when I express to you how a myelinated neuron conducts an electrical impulse. Down here at this end, this is called the terminus or terminal end. Or better yet, the axon terminal. And sometimes, depending on what book you read, these are called axonal buttons, terminal buttons. You can see why they're called that. It is in these little bulbs or buttons that neurotransmitter is assembled and stored in vesicles. So if you remember I'm going to just blow up one of these axon terminals. I'm going to magnify it. Within here are little bubbles with chemical in them, stored chemical. That's what's happening down here in this end. You just can't see it on my small drawing. Now, before I leave this, I'm going to use some terms that your book described. When this nerve cell, this is a typical motor nerve cell that we find in the human body. When it's not firing an electrical impulse, when it is at rest, it's not sleeping, it's manufacturing chemical, neurotransmitter. And how it largely does that is the raw material for that neurotransmitter is made up here in the cell body. I'm just drawing a mitochondria. Here are some ribosomes. There's an endoplasmic reticulum that has ribosomes on it. So this area, the soma body area of the cell, is making proteins, it's making raw material for chemicals, and then a lot of that stuff gets shipped down the axon inside of it, where we get to this point, and that chemical is assembled and packed, it's packaged, stored. Now, there's also material down here that needs to be recycled, or worn out particles that need to be disposed of. That material is taken back in the opposite direction to the cell body, where it can be broken down and recycled. That movement of stuff, transporting stuff to and from the cell body, is called axoplasmic transport. axo because we're in the axon, plasmic because we're in the cytoplasm of the axon, and transport. The two directional terms for axoplasmic transport are anterograde. This is all in your textbook, by the way. This means moving stuff from the cell body or soma to the terminal end. I'm just going to write terminal or terminus. Retrograde just means moving stuff in the opposite direction. So retrograde would be from the terminus to the soma, cell body. Okay. This doesn't have anything to do with firing an electrical impulse. It has to do with manufacturing the chemical that we store here that will be released when an electrical stimulus is generated. So I'm just looking at my notes really fast to make sure that I've gone over most of this language, and I have this. If you read or see this term, axon collateral, that just means a branch of the axon. So not all axons are perfectly straight like the one that I've drawn. It was just easy for me to do that. Sometimes they have branches emanating off of them somewhere like this, and we call those branches axon collaterals. All right, I'm going to say a few things now about this myelin sheath, this green stuff that covers the axon. First of all, not all axons are wrapped in myelin, have a myelin sheath. Not all of them do. Many do, but not all. Remember, in the peripheral nervous system, myelin is made up from Schwann cells, those lipid-based cells. In the central nervous system, myelin is made up from oligodendrocytes. This stuff, it protects, insulates. Let me go to a new page and just write myelin. It protects, insulates, and probably most importantly, not that the other two aren't important, it speeds up transmission of the electrical impulse. After we figure out how an electrical impulse is generated, I will talk about how myelin does speed that up. Remember, Schwann cells in the peripheral nervous system make up myelin, oligodendrocytes in the central nervous system. Remember that dendrites are never myelinated. Axons often are myelinated, but not always. Which brings me to two terms before I forget. These terms are largely used in the central nervous system: white matter and gray matter. The reason we call, if you were to slice open a human brain and look at it, there are spots or patches or places that look white. The tissue looks white in color, or the tissue looks gray in color. White matter, now that you know what myelin is, in the brain is myelinated, because myelin tends to be white, myelinated neural tracts, TRACTS. That means transmission pathway. Gray matter, when we look at brain tissue, are clusters of unmyelinated neural or neuron cell bodies. And this gray matter tends to be the processing information centers. So this is where we do most of our processing, AKA thinking and decision making. These are information transmission pathways that just carry an impulse from point A to point B. Last topic of this lecture is we're setting up. It's a long process and it is complicated. I'll do my best to simplify it for you, but we're setting up the discussion of how a nerve or neuron generates an electrical impulse. But there's some groundwork we have to lay down before we do that. So I'm going to write this term, membrane. You're going to read these terms in your book, and I want you to know what they mean. Sometimes the book does a good job, sometimes it doesn't, of explaining this stuff. This term, membrane potential, literally means that there is a difference in charge, C-H-A-R-G-E, I'm going to write polarity, on one side from the other. So along this membrane of a cell, if this is a cell membrane, membrane potential literally means that if we set up a situation where that the charge, the electrical charge on this side of the membrane is different than on this side, we've created the potential for something to happen, basically, for work to happen. That's why a battery that you buy at the store, I'm going to draw an old-fashioned battery here, has a positive end and a negative end. That battery stores potential for energy. It's an energy storage site. When we do that along a cell membrane, we also create sort of energy storage as well. So that's what membrane potential means. How this is set up along the membrane would take me about 20 minutes to really explain it to you well. So I'm going to do a really quick job of that. On this membrane, there are ion channels, and basically those are just proteins. There are proteins along here called ion channels that allow certain ions to flow through and other ions not to flow through as well. Most cells in the human body, inside of the cell, there's a high concentration of potassium ions, and outside of the cell, there's a high concentration of sodium ions. These ion channels, these leaky ion channels, allow a lot of sodium to leak out and not very much, a tiny bit, not very much sodium to leak in. So I'm just going to put a dotted line. The leakage of potassium, the constant heavy leakage of potassium outside of the cell helps create this positive negative environment, because we end up with a lot of positive ions out here, and taking away from the inside makes this negative, okay? This positive negative difference, this polar opposite, is called polarity, right? And to assist that polarity situation, there is an ATP-driven pump also on the membranes of our cells that keeps sodium out almost exclusively. It does pump potassium ions in, but then they quickly leak out as well. So I want you to get used to the word polarity. That means opposite charge, positive out, negative in. Membrane potential also means we've created along the membrane of the cell the potential for this cell to store energy and do work by making it polar. The membrane is polar. And finally, I'm going to write this. We've described what your book calls resting membrane potential. I'm going to go to the next page and write this out for you. because this is key for your understanding of the rest of this lecture, mostly next time, because we're almost out of time today. But I'm going to rewrite that: resting membrane potential. Now you know what some of these words mean. When the cell is at rest, across the membrane of the cell, there's a separation of charge that gives it the potential to do work. So this refers to a difference, or describing what resting membrane potential is. It's a difference of potential. In parentheses, I'm going to write ionic because it's created by those ions, potassium and sodium, across the membrane of a cell at rest. So this membrane, we say, is polarized. Let me write that. This membrane is polarized. That means that there's opposite charge from one side to the other. This occurs, this polarization occurs only along the edge of a cell. So if this is the outside of the cell, this is the inside of the cell. And for the reasons that I explained a few minutes ago, we have a positive charge out and a negative charge in. It's only along the edge that it's like this. It doesn't mean that all of this way out here is positive and all this way in here is negative. That's really not the case. It's just this separation of charge right along this edge. And it's because we largely have a concentration of sodium ions out here, and we have potassium ions in here, but they constantly leak out, and sodium is more positively charged than potassium is to begin with. It's a stronger positive ion. So at rest, I'm going to describe this situation to you. There's a distribution of ions along the edge of this cell, and that is sodium out, potassium in, the constant leakage of potassium out, and the fact that this is a stronger positive overall makes, that's why we just write positive, negative, but it's due to this difference. That's how our cells are at rest. And a nerve cell is like that at rest as well. So at rest, this neuron has that situation set up where we have positive along the outside of this membrane and negative right along the inside. And that occurs all the way around the cell. And we call this situation resting membrane potential, because this occurs when the cell's at rest and it has potential now to do some. I'm going to cut to the chase before I end this lecture. I'll start here next time with my next lecture. But just so you have an idea of what, so you're probably thinking, so what with all this separated charge and this potential, what's the big deal? Well, the big deal is just like with a battery. Here's a battery with a positive end and a negative end. If we connect these two ends, it causes a flow of ions from end to end. That's what creates some electricity, basically, is the flow of ions. And what we're going to find with this cell out here is when something happens to this cell, and I'll describe that something happening next time. we will get a flow of ions from one side of this membrane to the other, like this. And so we will get a bit of an electrical energy around this cell. And if this first change in polarity is strong enough, it will cause this part of the neuron to wake up and fire its own electrical impulse all the way down the axon. And then chemical will be released from these storage sites. So I'll go into those details next lecture. So let me cut my recording.